Welcome to our AMS Colloquium in a new fancy location here. Great to see everybody here today. So it's my very great pleasure today to introduce Dr. Victoria Booth, visiting us from the University of Michigan. And Victoria and I have been collaborating for over 10 years on various projects and sleep-wake modeling and various things, but today she's going to tell you about some work on neuromodulation of neural network dynamics.
Okay, well thanks. Thanks for the invitation. I'm glad to be here. I've been having a good visit the last 2 days, and so I realize this is a diverse audience. We've got the student visitors and people from different backgrounds, so we'll keep it informal, and so please, if you have a question, interrupt me. Don't worry about it. So I'd like to start by acknowledging my collaborator on this work. So this work is done in collaboration with Michal Zakowski, who's in the physics department at University of Michigan. And then in some of the different projects I'll show, I mentioned the different students who worked on the projects. Okay, so if we think about the brain is a network of cells, of neurons that are connected together by synaptic connections. But the power of the brain, what makes the brain so powerful, is that it's dynamic, it's got temporal dynamics and electrical activity, but it's also plastic, meaning that it can change. So it can change in terms of the actual properties of neurons can change, and the network structure of what neurons are talking together and how effectively they talk together changes as well. And so if we think about, so let's talk a little bit about the electrical activity of brain networks. It's caused by the electrical activity of the neurons in the network. And so here's a schematic of a neuron with the different parts, the cell body and the axon and the dendrites. And generally what the membrane around the membrane of the neuron maintains a voltage difference, and the electrical signaling of a neuron is a change in that voltage difference in what's called an action potential. So here's a profile of this sharp change in voltage and action potential on the scale of millivolts and milliseconds. And then in response to repeated stimuli or constant stimuli, it can generate trains of these action potentials. And so these action potentials, this voltage signal that neurons generate, it's transmitted to other neurons through synaptic connections down here, and it's that activity, the propagation of this electrical activity across neurons and across the network, that basically everything you do is somehow supported by this activity. Now I realized given the diverse audience, I'm missing a slide in between. So we think about, okay, so this is the biology of what's underlying the brain, and there actually is a very well-accepted mathematical modeling formalism to model the generation of action potentials, where there's a set of ordinary differential equations, nonlinear ordinary differential equations that can replicate these changes in voltage over time that an individual neuron can generate. And so that model is called the Hodgkin-Huxley model, and I shall show some of the equations a little later, but I realized I probably should have a slide here just to orient everybody. So we can build mathematical models of neurons, and then we can couple them together in networks of neurons. And so what we've been thinking about is, when you have a network of neurons, how do you know what the overall network activity is going to do? Given that each of the cells is going to be firing these action potentials, and they'll be sending the synaptic signals to each other across this network, what is the overall network behavior going to do? And so we've been building a lot of models, and mathematical models of these kind of spiking neural networks. And just to give you an idea of, a really simple idea about what kinds of network activity you see, I've got some movies here that show activity of networks activity of neural networks when we've got 2D networks. So here, each of the dots would be a neuron, and the edges indicate the synaptic connections between the neurons. And what I want to impress upon you here is that how the neurons are connected together will influence what the overall activity is in the network. So, for example, in the leftmost panel here, the neurons are coupled together just locally with just coupled to their nearest neighbors. And here in these movies, when the color is red, that means that the neuron at that pixel fired a spike, fired one of these spikes. So with the local connectivity, we get this kind of just propagation of activity across. But here, but what I'm going to change across the these panels, and especially in this right panel, is change the connectivity structure. So here in the rightmost panel, we've got random connectivity in the network, and in that case, we get a completely different activity pattern, where again it just started with one cell firing, but now the whole network is firing together. All the neurons are firing together. And if we choose a network connectivity structure in the middle, where it's not just nearest neighbor or not completely random, then we can get very complex kind of firing dynamics, where we've got a little bit of propagation, but then we get some more basically some more coherent activity across the whole network. So definitely the structure of the network, how the neurons are coupled together, which neurons are talking to which neurons, will influence what the overall network activity will do. We've been thinking about also, there's changes in the response properties of single neurons, and meaning how a single neuron will respond to its synaptic input. And one of the most drastic ways that individual neurons change their response properties is in response to neuromodulators. So neuromodulators are these very powerful chemicals that are manufactured in the brain and then distributed across the brain. And you've probably heard the names of some of these: dopamine, serotonin, norepinephrine, and acetylcholine. We've been thinking a lot about acetylcholine. And most of these are these neuromodulators, how they're delivered and distributed throughout the brain is usually there's some populations down in the different parts of the brain, sometimes in the lower parts of the brain, that manufacture these neuromodulators, and then they send their axons to all different other parts of the brain and deliver these chemicals, these neuromodulators, to the rest of the brain. And so these neuromodulators can have very strong effects on brain activity. And in particular, we've been thinking a lot about acetylcholine. So acetylcholine in particular, it affects arousal and attention. So high levels of acetylcholine promote attention. And acetylcholine is involved in cognition and memory. And it affects both cellular and synaptic properties. And so these neuromodulators, so basically throughout, just under normal conditions, these neuromodulators change their levels in response to different stimuli and also in response to different behavioral states. So for example, across sleep and wake states, waking and sleep states, these neuromodulators change their levels pretty drastically, where in waking there's high levels of all these neuromodulators. And it's actually thought that the state of waking, the alertness, the state of waking, is promoted by high levels of all these neuromodulators up into your cortical regions and in your upper brain regions. While during sleep, and in particular slow wave sleep, the non-dreaming states of sleep, the deep states of sleep, all those neuromodulators are low. They have low values of these neuromodulators. And so just as another sort of piece of evidence about how powerful these neuromodulators can be, basically all the drugs of abuse disrupt or modulate the levels of these neuromodulators. So for example, dopamine, it's modulated by cocaine and methamphetamines. Serotonin, the serotonin signaling in your brain is modulated by ecstasy, and basically many of the most popular depression drugs modulate serotonin signals in the brain. And then for acetylcholine actually, nicotine acts basically just like acetylcholine on your neurons. And so, all right, so sort of as a background of what neuromodulation is, and so what we've been doing a lot of mathematical modeling studies, numerical simulation studies, basically based on the question of how does the network structure influence the effects of, in this case, cellular modulation by acetylcholine to change network dynamics, or to not change network dynamics, as the case may be. So that's been our overall motivation, with the idea that if we have a fixed network structure of synaptic connectivity between neurons, but then we've got either high levels of acetylcholine or low levels of acetylcholine, how does the overall network activity respond to that change in neuromodulating levels? So just as a brief outline of what I'll be talking about. So first I'll talk about this cholinergic modulation of neural networks that contain both excitatory and inhibitory neurons and with different network structures, and then talk about a possible functional role for cholinergic modulation in sleep. All right, so let's look closely at what does acetylcholine do to an individual neuron? How does it change an individual neuron? And so what's been observed is that when in the presence of acetylcholine, the neurons, and in particular the cells that are called pyramidal neurons, so they're the main excitatory cells in your cortex and in your hippocampus, the main sort of parts of your brain that are underlying cognition, that in response to acetylcholine, they increase excitability, meaning they fire their action potentials at higher frequencies and more readily in response to stimuli. Another thing that, so we can see that in these experimental records where under no acetylcholine, the frequency is overall lower compared to with acetylcholine. Another thing that acetylcholine does is affect what's called spike frequency adaptation, meaning this transient change in frequency of firing of action potentials over time. Where with acetylcholine, you don't see it. So here without acetylcholine, you see this slowing down of frequency of firing, but with acetylcholine, you don't see that slowing down. The action potentials stay firing at a higher frequency. So these are two cellular responses, and another response or change of cellular properties with acetylcholine is that it affects its response properties to input, and these response properties can be measured in a what we call a phase response curve. So those of you who sat in the math bio lab meeting this morning heard about phase response curves, but I'll talk about what a phase response curve is in terms of neurons, in terms of neuron firing. So actually, phase response curves came from mathematical theory. They came from the theory of mathematical theory and mathematical analysis of oscillating systems. So, and only recently have they been applied to neural firing to try and to basically categorize or characterize neuron responses. So, in the context of a neuron, what a phase response curve measures is if we have a repetitively firing neuron, so a neuron that's firing action potentials, and we'll assume that it's firing periodically with a regular period. The phase response curve what it measures is the response to the timing of firing if the neuron gets a stimulus at some point during its periodic cycle. So for example, in this case, the stimulus arrives here, and this dashed line shows what is the change in the voltage trajectory over time in response to the stimulus. And you can see that there's a change in the timing of firing. So what the phase response curve does is actually measure this change of firing as a function of when this stimulus arrived, at what time in the cell cycle this stimulus arrived. So the plot of the phase response curve will be on the x-axis will be the phase of the periodic cycle of the neuron and the timing of firing of the stimulus, and the y-axis will say what is the shift in timing of firing. And what we usually do is, instead of plotting it as a time shift, we'll plot it as a fraction of the period, so a phase shift. And in this case, the timing was delayed after the stimulus. The neuron fired at a later time, so we just plot that as a negative phase shift. And so, for example, but some neurons show, like, if depending on the timing of firing, the effect of the stimulus can be different. So here, with this neuron model, when the pulse arrives later in the cycle, the timing of firing is actually advanced, so we get this shape with positive phase advance later in the cycle. All right, so well, what's been observed under acetylcholine for cells in the cortex, these pyramidal cells in the cortex, is that the acetylcholine can change the profile of this phase response curve. So here are some experimental measurements of phase response curves. Okay, so these are really noisy, right? Yeah, super noisy. But basically it's hard to measure a phase response curve in an actual neuron where you're stimulating them. So these are the results of many experiments where they're perturbing, or delivering brief stimuli to repetitively firing neurons. But generally, okay, so they fit a line between it. But you generally see a difference between when there's no acetylcholine and when there is acetylcholine. And what they notice is that when there's not acetylcholine, generally stimuli earlier in the coming in earlier in the phase overall cause a delay, or later in the phase they call it advance. But in presence of acetylcholine, that delay region is diminished, and where generally, overall the mean behavior is an advance. All right, so it turns out that this distinction of profiles of phase response curves, from phase response curves where there's maybe an initial delay period, or where the advance period is skewed to later, or the advance phases are skewed to later in the period, compared to phase response curves that are more symmetric in terms of and predict advances in timing of firing across the cycle. That those are are categorized as two different types of phase response curves. And they came up with these very original names, where this profile is called a type 2 and this profile is called a type 1. All right, but so we can see this difference in the phase response curves with acetylcholine, but why should we care? Why would they care? Or why does it matter? Well, so actually, the phase response curves, what they can predict is how the timing of firing of a neuron, so how a neuron's firing is going to respond to the input it can get. So in a way, we can kind of extrapolate to how an individual neuron embedded in a network is going to respond to network activity, or how it's going to respond when neurons are coupled together. So it gives us a little bit of insight, some insight into, if neurons are coupled together with specific types of phase response curves, how are they going to respond to each other? How are they going to fire, say, or relative to each other? And it turns out, so as I said, phase response curves were first developed in terms of coupled oscillator theory. So there's been a lot of mathematical analysis of phase response curves, and there's been a lot of mathematical analysis of these phase response curves that come from these type 1, type 2 phase response curves that neural models show. And there's been some results, some analytical results that show and prove that neurons with type 2 PRC profiles, they synchronize better than neurons with type 1. So here's why they would care about a PRC, is because it could, if this math theory is right, it would predict that if we've got neurons with a type 2 PRC profile, when we put them in a network, they should synchronize. They should synchronize better than if they were had type 1. So and we can understand intuitively, without getting too detailed into the math theory, why neurons with a type 2 PRC would synchronize better than type 1. All right, so I've got some kind of cartoon schematic about that to give that explanation. All right, so let's think about two cells, the blue cell and the red cell. And the blue cell is firing and sending a synaptic input to the red cell. So we talk about the blue cell being presynaptic and the red cell being postsynaptic. So if these lines, the blue lines and the red lines, indicate the timing of firing action potentials of the blue and the red cells, let's consider some different cases of relative timings of firing of the two cells. So whether we have a type 1 or a type 2 PRC. So here, let's see if we think about the blue cell is firing, and the red cell is receiving the synapse. In this case, it's receiving the synapse just before it wants to fire itself. So it's receiving the synaptic input late in its firing phase. Both the type 1 and the type 2 PRCs predict an advance in a phase advance in the timing of firing, meaning earlier timing of firing. So in that case, so after this input, the next time the red cell should fire, it should fire a little closer to when the blue cell would fire. So in that case, we can definitely say, okay, we're going to get some synchronization, that the neurons should start to fire a little bit closer in time. But what about if we have the opposite sort of phase of firing, where the red cell, it fires its spike before it gets the input from the blue cell. So in this case, we can think about that the red cell is getting the input from the blue cell early in its firing cycle. And so for a type 1 neuron, that'll advance the timing of firing, so actually make the red cell speed up its firing, and so that the next time they fire, they're going to be firing farther away from each other, not closer together. But with the type 2 PRC, where if the red cell gets the input from the blue cell early in its firing, it'll actually delay the timing of firing. So in that case, the next time the red cell fires, it should fire closer to the blue cell, so it's a little more synchronized. So basically, with this type 2 PRC profile, where depending on the timing of firing, the neuron can speed up its firing or slow down its firing, they synchronize better because they have more flexibility in their timing of firing. All right, so this was very intuitive, but as I said, and then there was math theory that predicts this. But the math theory was really done only with 2 cells, and we just did this explanation with 2 cells. All right, what's going to happen in a big network, where you've got a lot of cells coupled together, and neurons are getting not just one input every cycle, but multiple inputs every cycle. And the network may be structured in different ways, like the synaptic connection topology would be different in different ways. Does this hold up? Does this change in PRC profile, which we said acetylcholine can change, so high acetylcholine gives us type 1, low acetylcholine gives us type 2. Will the change in acetylcholine and this change on the cellular level actually translate to changes in synchrony across the whole network due to the cellular change? So that's what we've been investigating that in different types of neurons. And so one of our first studies, this was an undergrad, Andrew Bogart, who did this study. We built some big neural networks where the individual neurons, here's our schematic of the neuron with a cell body and a dendrite, and with we use this Hodgkin-Huxley formalism here. I don't show the equation, but I'll show it later, where the individual neuron can generate this repetitive firing of action potentials. And we basically tuned the parameters, tuned basically the ionic currents in the model, the different ionic currents in the generating the action potential in the model to generate 4 different profiles of phase response curves, where some of the profiles were more type 2-like this orange one and the red one, and some of the profiles were more type 1-like, meaning they were still a little bit skewed to the right, but not as skewed as the orange and the red one. So the blue PRCs are more type 1-like, and the red and the yellow ones are more type 2-like. And so we built networks containing these model neurons, and we wanted to consider different network structure topologies. And so what we used is a small world network paradigm to systematically vary the network topology from a local connectivity structure, nearest neighbor connectivity structure, to a random connectivity structure. And so how we can do that using this small world network paradigm is we can start with a locally connected network where the dots are our neurons and the edges are our synaptic connections. So start where our neurons are only connected to some of their nearest neighbors, and then define a rewiring probability parameter P, whereas a probability of an individual synapse being rewired somewhere randomly in the network. And so when we increase the rewiring probability, some fraction of the synapses will be rewired randomly in the network, and eventually when we make the P equal to 1, then we're at a random connectivity. So in this way we can systematically change the network structure and kind of scan all the possibility, or some, you know, across different network structures. All right, so what did we find? We basically found, so this is a big summary we did a lot of numerical simulations of all these different types of networks, where what we varied was, let me go back a second. What we varied was this P, this rewiring probability for all the networks, and we also varied the number of synapses, the number of outgoing synapses per neuron. So how dense was the connectivity? So we had that parameter, we called it R. And so this is basically a summary of all these numerical simulations of these networks of 250 neurons coupled with excitatory synapses, where in these panels up on the top, they're like two-parameter bifurcation parameters, where we've got our value of R varying on the y-axis, so the number of synapses outgoing from each neuron, and the value of P, our rewiring parameter, going from 0 to 100% on the x-axis. And what's shown in the different regions here is what kind of network behavior we got for the networks containing the different cell types. So where A, B, the cell types A and B were more type 1-like, and cell types C and D were more type 2.
So are all the cells the same in each experiment?
Pardon?
So when you have cell A, does that mean that all the cells are cell A?
Yeah, there's some heterogeneity in the cells. A little bit of, maybe they didn't have exactly the same firing frequency. A little bit of heterogeneity, but generally, right, all the cells had that same profile. Exactly. And so to see what the overall activity pattern is, what we're plotting here are raster plots, where we've got time on the x-axis, and on the y-axis is the neuron number, the index of the neuron, and whenever there's a dot, as you move across horizontally, whenever there's a black dot, it means that neuron fired. So it's a way to visualize the overall network behavior. And so, all right, so the takeaway message here is that these networks that had the type 2, more type 2-like PRCs, they really had only a limited type of dynamic that they showed, where most of it was what we call this network bursting, where here are the raster plots for network bursting, where all the cells are basically firing at the same time, or in a very short time window together, so basically synchronously firing. And so for this large regions of parameter space of network structure, these type 2 kind of cells want to go into this bursting, want to go into this synchronous bursting. While these neurons that have the type 1 PRC, they're in the same network structure, or the same, across the same wide regions of network structure. They want to do different things. They don't want to necessarily all synchronize. And so in this way, it's the cellular property that's allowing or permitting or providing for this different variability in network activity. So that's one takeaway. The other takeaway is that eventually there are network structures that will constrain what the network is going to do. And so down here in the right corner of all these networks, basically we've got a lot of synapses, so very dense connectivity, and we've got the high P, so a lot of random connectivity. So the networks are really well connected and connected randomly across it, and then that promotes bursting no matter what the cell type is. So in this way, what we see is that the network structure can constrain network activity, where all in this upper right corner, all the networks are showing this synchronous bursting, but there are network structures where the cell type can have a big influence. All right, so so this was all networks with just excitatory neurons, but in brain networks, there's neurons that send excitatory synaptic projections to each other, meaning when they fire an action potential, they tend to excite the other neuron. But there are also neurons that send inhibitory projections, meaning when one neuron fires, the effect of its firing on the downstream neuron is to suppress the firing, so inhibitory network. So here's just a cross-section of an area in the cortex where these pyramidal cells are the main excitatory cells, there's some excitatory cells here, but there's lots of these little tiny other neurons, and those are usually inhibitory. So that for most of your brain, there's both a mix of excitatory and inhibitory cells connected together in some way. And in particular, some of these inhibitory cells can be modulated by acetylcholine, and have shown the same kind of cholinergic modulation that I showed you before, which was shown for excitatory cells, meaning changing their PRCs and changing their excitability. So this table just talks about all the different types of inhibitory cells in the hippocampal networks, and whether or not I just indicated which ones are cholinergically modulated. All right, so now we're going to talk about excitatory inhibitory neural networks. And in this case, what we'll use for our cell model is a Hodgkin-Humphrey type model that we can more directly simulate the effects of acetylcholine. So here's the main equation for the Hodgkin-Humphrey type neuron, where we have a variable V for the voltage in an individual neuron. And for those of you who are familiar, we've got a sodium current term and a potassium current term, a potassium delayed rectifier current term, and a lead current term. But we also have this additional potassium current term, which is called an M-type potassium current term, which is a slow adapting current that is shown in the different in pyramidal neurons. And what acetylcholine does is it actually blocks these M-type potassium currents in individual neurons. So we can simulate the effects of acetylcholine by just turning off this GKS, the strength or the conductance of this M-type potassium current. And we end up with what it can do. It does change the excitability of the mononeurons. So I'm not showing that, but it has a big change on the PRC. So this model, when we change this parameter GKS, we can switch it from a type 2 PRC when GKS is high to a type 1 PRC when the GKS is low to simulate the effects of acetylcholine. All right, so we've been doing some simulations in excitatory inhibitory networks, and so what we're going to have is we're going to have a population of inhibitory cells or I cells, and a population and network of excitatory cells and E cells, and the I cells will be connected to each other, and the E cells will be connected to each other, and then there's connections between the I cells and the E cells. All right, so we've got this big network here. We're going to have 1,000 cells, 800 are excitatory, 200 are inhibitory, with random connectivity, and we're vary the connectivity density between 10 and 30% between these populations. So what we've been thinking about, we'll vary, and we'll also vary the connectivity strength. Where what we've been generally thinking about with these kind of EI networks is we'll think about varying the strength of the interconnectivity between the E and the I cells, the excitatory and inhibitory cells, and vary the intra-connectivity. So the connectivity within the E cell network and within the I cell network. So in this structure of excitatory inhibitory neural networks, there is a regime where what's known as where this synchronous network bursting is promoted depending on this network connectivity. And basically this regime is when there's high interconnectivity between the E cells and the I cells, a type of synchronous network bursting that's called ping can happen. All right, I'll give you a little primer on ping, synchrony. So ping stands for pyramidal interneuron, so pyramidal cell inhibitory interneuron network, and gamma. And gamma refers to a frequency of synchronous firing across the whole network. And how these E and I cells tend to synchronize each other actually depends not so much on what the E cells want to do on their own, but it's more that the inhibitory cells, the I cells, gate the timing of firing of the E cells. So here's a cartoon that kind of shows how this synchronization can happen between some excitatory cells, which are green, and some inhibitory cells, which are red over time here. And then what's shown on these traces is the overall synaptic signal from E to I, so from excitatory cells to inhibitory cells, and I to E, the opposite way. But the idea of this, how the thing synchronizes, is that if the excitatory cells are firing, and enough of them fire to cause inhibitory cells to fire, when the inhibitory cells fire, they send we can think about a global inhibitory signal to the excitatory cells and suppress all the excitatory cells. And so the excitatory cells are going to be quiet, so here they're not firing, and they can only fire again when the inhibition wears off. So if this inhibition is kind of long-lasting, it can suppress the excitatory cells for a while, and then it wears off, and once it wears off, all the excitatory cells are ready to fire again. And so then they fire again in closer proximity. But when they fire, then they make the inhibitory cells fire again. And the inhibitory cells then suppress all the excitatory cells again. And then the next time they fire and they're released from inhibition, they can fire again closer together. And so it's actually thought that this is a very robust way in brain networks, especially cortical networks, to generate synchronous firing, with this gating of timing of firing of the excitatory cells by the inhibitory cells. All right, so I'm bringing this up because it turns out to be a constraint network structure that will dictate what the network will do. Depending on, or as we'll see, in some cases, regardless of whether the cells want to fire together or not. So here's some studies by Scott Rich, a graduate student he's actually defending in 2 weeks, where he was doing these simulations in these E to I networks and simulating cholinergic modulation of the E cells and the I cells in all possible combinations. And basically, here's again is a cartoon of our overall results in terms of if, depending on network structures where we're varying interconnectivity or varying intraconnectivity. What we found is that if, so up here in this upper right-hand corner, that's when the interconnectivity is strong, but the intraconnectivity is weak, then the acetylcholine changes to the cell types had very little effect. And in fact, this is a regime where all networks, no matter what the cell types were they type 1 or type 2, whether excitatory inhibitory cells type 1, type 2, we always got this synchronous network bursting, which we could do to this ping mechanism. Where here the E cells are the green cells and the red cells are the I cells, and basically the E cells are always firing synchronously. Compared to, let's see, so this other region down here where if the interconnectivity is weaker, and no matter what the and with varying intraconnectivity, then the cell type, what the individual cells wanted to do, had a big effect on whether or not we got synchrony. Where, for example, if the excitatory cells were type 1, meaning they don't synchronize as well, they wouldn't synchronize as well. But if the excitatory cells were type 2, then we would get this synchrony of the excitatory cells. No matter, it didn't matter what the inhibitory cells did in that case. And so we did so Scott did a lot of, again, parameter scans across interconnectivity strength and intraconnectivity strength. And so we're here, each pixel represents one of these different network structures. And in this case, what we're measuring, the measure we have, so the color is a synchrony measure. So how synchronous is the firing of the excitatory cells or the inhibitory cells, depending if what type of PRC the individual cells had. And so what we can see when the excitatory cells are type 1, no matter what the inhibitory cells are, the synchrony only sort of happens in this upper right corner, where that's the ping regime, where we can say, okay, it's really network structure that's dictating synchronization in this case, but all in the other regions of the network, different network structures, the type 1 cells wouldn't synchronized, in contrast to when the excitatory cells were type 2, then we got red hot colors in all the, basically across all the network parameter regimes, meaning that we get this synchronization of the cell. Due to, in this case, we could say, in a lot of the areas, due to the cellular properties. All right, so just to finish off the effects up in this other corner where we've got basically high inter and high intra connectivity. There it was a little more subtle in terms of what were the effects of acetylcholine, where sometimes even with the type 1 cells could synchronize, but it depended on what the inhibitory cells wanted to do. If they synchronized well, or if they didn't synchronize so well, then the type 1 cells wouldn't synchronize. So it was a little more subtle up in that. All right, so in this case with random connectivity between the E and the I cells, the network structure, we got different we could say the cholinergic modulation had differing effects on what the overall, whether or not it changed the overall network structure. So there's in other parts, in some brain networks, so that was random connectivity between the E and the I cells. In parts of the brain, there's a more structured connectivity structure between excitatory cells and inhibitory cells. And in this work by another student, James, we looked at what's called a center-surround connectivity structure, where we've got an excitatory network, and here we were doing 2D networks, so an excitatory network of blue cells and a network of inhibitory cells in red. And the connectivity structure was such that the excitatory cells only connected to their local nearest neighbors, other excitatory cells, and only locally to the inhibitory network. In contrast, the inhibitory cells projected globally to make synaptic connections globally across the excitatory network and globally across the inhibitory network. So we've got global inhibition and local excitation. And so what was interesting here is that we got a different type of effect of acetylcholine, what acetylcholine did. And so what we found is that in the high acetylcholine case, what this movie shows, here the color is the firing frequency of the cells in the pixels. So this is the 2D network, and when it's red, that means the cells were firing. So with high acetylcholine, we would get this stationary bump of activity that would kind of stay, maybe it'll wiggle around a little bit. But in the low acetylcholine case, we got traveling activity of the bump. So here what we have is we've got the periodic boundary conditions on our network. So we've got one bump, but it's periodic boundary conditions. So a bump that's moving across, a bump of that firing activity that moves across the network. And so this was like a completely different type of pattern, activity pattern, because of the different connectivity structure. But what was even more interesting is that without going into a lot of details, but what was more interesting that in this case, it wasn't really the PRC that, I've been harping on the PRC this whole time. It wasn't the PRC that underlined the difference between a stationary bump and a traveling bump. What it was the cholinergic change to the spike frequency adaptation. So this change in firing frequency, whether or not a neuron changed its firing frequency over time, it was that change in spike frequency adaptation with acetylcholine that dictated whether or not we got a stationary bump or a traveling bump. And we can kind of understand it is that in the case with no acetylcholine, if the inhibitory cells had this spike frequency adaptation and their firing kind of slowed down, it let the excitation propagate through the network. It let the excitation move. But if the inhibitory cells fired at a maintained firing frequency, it kind of corralled the excitation and wouldn't let the excitation propagate across the network. Okay. So the PRC did actually, though, have a subtle effect, a minor effect, and it was mainly in what was the timing of firing of the cells within the bumps, the activity bumps, where with the high acetylcholine case and the type 1 PRC, within the bump there was, here is showing asynchronous firing of spikes within the bump, compared to in the low acetylcholine case with the type 2 PRC, there was more synchronous firing inside the activity bump. And these are histograms of the relative phases of firing within the bumps for different levels of the acetylcholine. All right, I'm running out of time, so I'm going to skip the summary, because now you might wonder, all right, so I've been telling you about all these, basically numerical studies, just trying to pick apart and understand what is a network going to do, but why would it matter? Why would, does this happen in the brain? Does this help in the brain? Does it have an effect? Well, it could perhaps, what we want to propose is it could have relation to one of the reasons why, or one of the leading hypotheses about why we sleep. Right, so it's actually pretty amazing, given that basically every animal, every mammal for sure, and most animals have a rest period, have a need to sleep, have a need to rest, but it's not, we don't have a full understanding about why. Why is it that we need to sleep? Now, one of the leading hypotheses is that we need to sleep to reset the synaptic connections in our brain. And so this is called the SHI hypothesis, or the synaptic homeostasis hypothesis, and it's mainly promoted by Tononi and Sorelli from the University of Wisconsin. And the idea is that when we're waking and we're learning, we're getting a lot of stimulus and we're learning new things, how learning happens in the brain is that the synaptic connections between neurons strengthen. They get stronger. And so with learning, they get stronger, and so there's the process called synaptic potentiation, where the synapses are stronger. And so stronger synapses are actually physically bigger in the brain. They actually take up more space. And so when we have, if you think about if we just kept learning and our synapses potentiate, we'd have these really big synapses across all of our brains. So we would run into issues of space. We've got, our brain is constrained by our skull, space costs and constraints, as well as energy costs of maintaining these physically bigger, cellular parts that are with the synapse. As well as, you can think of saturation of the network, because if all your synapses are potentiated and strong, there's no room for learning something new, for strengthening of some other synapse. So this state of high potentiation is not sustainable for a healthy brain network. So the idea is that during sleep, there must be some renormalization, or there should be, put it that way. There should be some renormalization of synaptic strengths where synapses get weak again, so that during sleep some of the synapses weaken, so that by the time we're after sleep and we're ready to wake up again and learn, there's room in our brain to learn. So there's been a lot of studies. So this is their hypothesis, but this is really hard to show whether or not this is true in the brain. And so they've been doing a lot of experiments trying to show this, and they've got some evidence that it happens, but they don't have a really good explanation about how it could happen. How could this happen? That we get this synaptic, say, downscaling or rescaling or renormalization in the brain. One hypothesis is that it could be due to this change in neuromodulator level across waking and sleep states, where I was telling you before, where lots of the main neuromodulators are high during waking and they're low during sleep, including acetylcholine, our favorite one. And so to investigate that, what happens in a network? So we know when we change acetylcholine, we can change network dynamics. And if we change network dynamics, how does that change how synapses would strengthen? How would they change their strength due to their different firing patterns? And so we did some other studies with a student, Chris Fink, in, again, excitatory inhibitory networks, randomly sparse, where we modeled it with that same cell, the neurons with the same cell that could change PRC types with acetylcholine, but now we let the synaptic weights change in response to the activity patterns. And one of the leading rules or leading mechanisms or how synapses change relative to their timing is called spike timing dependent plasticity, or STDP. And what they've shown experimentally is that depending on the timing of firing of two cells that are connected synaptically, the synapse will strengthen or weaken. And so this is the experimental results where we've got the timing of the postsynaptic cell and the presynaptic cell. And what they observed is that if the postsynaptic cell fires after the presynaptic cell, then synapses strengthened. But if the postsynaptic cell fired before the presynaptic cell, then synapses weakened. So depending on the timing of the firing, and it was only happened, oh, I don't have the time scale in here. This should be milliseconds in here. So it only happened within a small millisecond window of the timing of firing. So they had to fire within close proximity and in some order, and depending on the order, they strengthened or they weakened. All right, I forgot, I know I'm probably doing University of Michigan time where I think I have until 4 o'clock, but I probably don't have till 4 o'clock, right? Is that right? Am I running out of time?
Time for questions, but yeah.
All right, okay. All right, well, so I'll be real quick. So again, so we built these networks, we varied network connectivity. All right, so basically here we've got networks, the type 2 networks where we had synchrony, like before, and the type 1 networks, acetylcholine, where we had asynchrony. So we've got changes in synchrony because of the PRC, but what does that do to synaptic potentiation? And we measured synapses by basically looking at histograms of the overall synaptic weights after the network had simulated for a while. And what we found is that, so here, this measure of synaptic skew measures the, just put it all out there, when we had this asynchronous firing in simulated high acetylcholine, so simulated waking, we could get overall potentiation of synapses. So here's a histogram of the synaptic strengths, get overall potentiation. But when we had this synchronous firing, say in low acetylcholine cases, like say simulated sleeping, what happened is we got this bimodal distribution of synaptic strengths, where some of the synapses were potentiated, but some of them weren't. And if we switched the acetylcholine level up and down as would happen between waking and sleeping states, we could get this potentiation during waking and then this sort of renormalization during sleep. All right, so since I'm out of time, I'll skip this part and then I'll just, okay. And so I'll just end. Acknowledge my collaborators, Mihaly Zakowski again, and then the different students who worked on this project, as well as the funding. Thanks.